trafficking into the microenvironment. So in this particular case, we are giving a one dose before the actual treatment. Right? It could be chemo, could be pembrolizumab. We do see some side effects, osmosis, diarrhea, fatigue, and it's very manageable.
Thank you. So the second question is from Prof. Chin. So I'm very interested in the primary tissue and how to get them for future research. So what is the procedure to get the primary tissues from your organization? So like right after the patients pass away and we are contacted, or?
Thank you very much for these very important questions. Our institution actually has a committee, Science and Research Advisory Committee, as well as an independent ethics board. So basically, if you would like to collaborate with a center, what you can do is that you can basically contact us, write to us about your proposal, and then we will go through our science committee, and the chairman is Prof. and then we will go through with our committee members, and then if approved, then we will then release the tissue to you. It is a prospective study whereby you tell us what kind of tissue to get, and we will procure the tissues for you. Then we will set up informed consent for our donors so that we can explain to them that rather than just repetition, of course we have consent for repetitions, which then they actually donate the tissues and let us decide whatever research that we want to do. But it's also important to know that, to let them know that if there is an existing project that specifically look for specific type of tissues for specific research, that will be very good, because they will know that clearly after they pass on, their tissue will use in that direction. So I think that would be an excellent example of collaboration if we can have this setup. Yeah.
Thank you, Prof. Chin.
We are actually a non-profit, so we don't sell tissues through collaborations. Yeah.
Okay, thank you very much. I think I have a question for Dr. Akhmal. So my question is, Malaysia is often seen as a relatively small market, and in some cases the cost of conducting trials can be relatively expensive with our regional neighbors, especially in oncology. So in the context of oncology, what do you think is the biggest challenge in getting the big pharma, big players to come to Malaysia? And how is Malaysia positioning itself with all the things that are happening with the competition and everything?
Like I said earlier, back in 2015, it takes 350 days to conduct clinical research in Malaysia. And we have changed that to become 90 days. And that is one of the key value proposition that attract sponsored research. Now for oncology, it's a different ball game altogether, because not only we have to deliver with speed, reliability and quality, we need to have one investigator. It doesn't help sometimes when investigators are moving from one sector to another, because the interest can change. Number two, study coordinators. I think when we do this, we know that we manage to have a pulling factor for people to stay in the certain sectors, and as a result, we get more studies. Now, these are not cheap studies. They're very expensive. Having said that, it is a cost-effective where we are competitive with other nations. Does that answer your question?
Yes, it does.
So that actually attracts more study and some key obstacles that we face.
So to summarize, the track record proves itself and you get more people to come in. So yeah. Thank you.
Let me tell you this, when we first started the first incumbent, attracting a pharma company is a huge challenge.
Indeed.
We only managed to get a small, medium-sized pharma, which actually work with their own baby compound. And because of that success and the publication in the AACR, it helps to open up the eyes for other pharma. And I'm very grateful to that.
Thanks, Dr. Akhmal. Okay, do you all have any more questions?
I got one question for.
Of course,
Prof.
of course.
Yeah, I'm not prof either.
No.
But the thing is, what you do is very impressive, and I just wonder whether that has been a very good step in terms of understanding how smaller dose can help in Asian patients.
This is actually very, there's no pharmaceutical paper here, right? I'm very careful with that. Yeah. So the hard truth is that
A lot of them are actually expired in terms of their patents. So
in fact, it's true because we are actually seeing, for example, in the case of BEF, we actually found a parallel trial with sunitinib, right? And we realized that the better response actually dose at the lower dosage, right? And this is not just a single dose trial. We have multiple trials that show the same thing. That if we have a dose escalating trials in which we try and lower it, we often get a better response, right? And the hard part is really how do we actually make the claim, because the indication from the pharmaceuticals to us in Singapore, we have Medishield, right? So if it's a paper, we have a specific indication. You must finish, right? Everything we did is for it to be claimable, right? And that becomes a big challenge because if you go against pharma and say, hey, I'm going to reduce the dose of my patients, are you going to sponsor it? They'll say no. They say this is basically making me lose more than half of the money. Of course, I'm not going to. So there's really a fine line between reducing drug doses. But what we are trying to do is, like you say, we are targeting those drugs that is past the patent shelf life. We are lowering the dose, for example, Avastin in our case, right? We have actually 4 or 5 different trials using Avastin as a way to improve treatments in lung, in hair and neck. That is something we can do because they are no longer being protected by any patents or any companies, right? But for those new drugs like pembrolizumab, we can't. We simply can't. ADC, we simply can't. Right? This is hard truth about the drug business.
Unless you sponsor it yourself.
Yeah.
I mean, it's not that you can't.
Yeah.
You just cannot get pharma-sponsored trial.
Yeah. It is really funny with that statement because for this
From Singapore.
Yeah.
for this, so Bunche is a very different idea from here, right?
Yeah.
If let's say we have a trial, it works, and eventually it's not going to be approved by FDA, then what's the purpose of doing that? Right? So we always try to run the trials that we know for sure the pharma will support it, right? This is one example for this. So MSD funded the entire BEF, Avastin, and pembrolizumab trials. So they give up halfway because their drug alone is not working. So they just give up halfway, say, hey, I'm not going to fund it anymore. So of course we finish everything, with our own funding and everything. And now because this is a population cancer oncology, FDA may approve it as a center of care, right? Now MSD is coming back and say, how we can support the next studies, right? So it's a very realistic balance of what we can do to attract their attentions. Right.
I'm glad you're in the forefront for this. Thank you so much.
Trying to do some changes to the field. Right.
I have a question for each of the panelists. Dr. Akhmal, can we get a comprehensive list of the trials ongoing in Malaysia?
Can you use the mic so that everyone can?
Right, Emma1.
Yeah, the short answer, you can just let me know what you
So
need.
do we have it available on your website?
we do.
Things like that?
Without revealing the company, I don't think there is any issue. The slides just now, I'll be happy to share with you because that gives the comprehensive of what happens in 2024. Huge number of what they call compound. Asha is back there. She'll be the contact point.
Yeah, so we'll talk to you about that. Dr. Kong, I wanted to ask regarding your experience in Singapore. How big of a biobank do you have in Singapore, and how many patients do you need to drive the machine learning that you talked about?
Very interesting topic because Singapore always come to the same challenge that we don't have enough numbers. With 6 billion populations, you can't say much. But what we are really trying to sell is that we of course we have the precise cohort. This is really just 10% of our population maximum that we can provide. And this is just healthy blood, just like UK Biobank and Genomic England. This is healthy individuals. There's no disease correlations. But when we talk about biobanks, we individually within the different medical classes, we do have a biobank, right? We are always trying to improve that. So we call it tissue repository, right? So this is really to support acquisition of samples. So in the old days, it's only just FFPE. But now because we have more single cell, more spatial, we also take OCT, we also improve the guidelines, right? And in my lab, we actually have a clinical protocols. Like we identify certain individuals that we want. Let's say we talk about cancer progression in lung. We know these individuals coming in, right? We actually have a mini biobank within our lab, but those are very specific to our research purpose. So we have a larger one that's a hospital-based, right? Singapore as a whole has discussed the need to have a biobank, but the argument is that with the small population, is it going to be able to compete, right? So we started having all the progressive serial collection of samples. So we can't compete in terms of the baseline. The number, if everybody's a cancer patient, is only 6 million, right? We still can't compete with China, with other bigger populations. But what we have is the serial time of every single individuals. Every time the patients come into the clinic for check, we take the draw bloods. Every time the tumor relapse, we constant that we put in the effort to constant them for biopsy. So we actually make sure that for every single patient, we trace and track every single evolution. The point of relapse, we actually have that. The point of treatment with the plasma. So this is something we actually do it.
I think that is very useful because you have at the different time points. Therefore, it's easy for you to see the progression, as opposed to having it only a diagnosis and you don't have valuable data from that. Thank you. Prof. Chin, I wanted to ask, is there a possibility of, like, if let's say the patient consent into the rapid tissue donation program and to have the primary clinicians harvest the tissues upon the death of the patient? This is because most of the patients I see in my practice, they kind of have this rapport already with the primary clinicians, and they may feel that it's better for the primary clinicians to harvest those tissues because they feel that those clinicians understand their disease more. Thank you.
Yes, I totally agree with that. That's why we talk about primary physicians having a rapport with the patients. And of course, it depends on the facilities for the harvesting. So if the donor site is literally a facility for harvesting, what we can do is that we can facilitate that. And then we can, because nowadays we also have a freezer truck. So our truck can go anywhere in Malaysia. So what we're thinking about is that, if, for example, in Penang, if there's a center that can harvest the tissue in Penang, then our donor can go there for harvesting, and then upon harvesting, then we can quickly freeze the body for the supplemental program. So this is what we can set up at this moment.
Thank you, Prof.
Now, of course, if we have ENT patients coming to our center, we need your help.
No problems.
Thank you.
Okay. Thank you. Do we have more questions? Okay.
Hi, I'm looking from IMA. I would like to ask a question to Dr. Kong about the NPC study. Since anti-VEGF-RA is about anti-angiogenesis and anti-PD-1 is about the unblocking T-cell killing, have you ever explored into analyzing the treatment, the diagnostic issue to identify responder and non-responder?
So we actually have a paper in Nature Cancer, it's under review right now. So we're trying to look for the specific changes. So we know that VEGF will actually induce T-cell infiltration or B-cell infiltration into the tissues. So what is actually doing to endothelial cells? Because we know that for VEGFR is specifically expressed on endothelial cells. So we actually have papers and try to understand within the endothelial cells and the fibroblasts around those, what are the key changes after bevacizumab treatments? And what are those key changes basically alter the microenvironment? So this is actually under review now, but I can't really say much, but yes, it's a good question, so we're looking into it.
Perhaps the next question from a non-clinical point of view, we always wonder what makes you decide what available drugs can be explored in a clinical trial? Why would one decide to combine radiotherapy versus anti-VEGFR or immunotherapy versus chemo? So what is the factors that lead to consideration of oncologists to try this combination in clinical trial?
I really think this question really, Prof. Chong is the person to address. But in my own opinions, I think firstly it's also depending on the availability of funding. So who is funding? If the farmer come to you, you have to stick with what they give, right? But from an investigator perspective, when we initiate the clinical trials, we always want to do one step at a time. I'm not going to try 2 different drugs at the same, right? So for example, in this case of the BEV studies, we started by giving BEV to chemo-RT, which was then the center of care for nasopharyngeal cancers, right? We give BEV, we combine with chemo-RT, it works. We get the paper out, we move on the next step. Can we give BEV now with pembrolizumab? It works. We actually have a 3rd phase coming up, in which we are giving pembrolizumab, BEV, and chemo-RT, right? And the result is even better. So we're always taking it one step at a time, right? How can we advance? How can we do it? The tumor is always going through progressions. So how can we design the trials to better treat that, right? So for chemo-RT, we see that it works very well for chemo-naive tumors, right? For pembrolizumab, we see that it works very well for metastatic tumors, right? So if you combine that PD-L1 inhibitors with chemo-RT, does it actually give a agnostic kind of way to all different stages of nasopharyngeal carcinoma? It's something we're actually addressing now. So there's really no quick answer to your questions, but it's really progressively we understand the field, what is available, and how to advance it.
Thank you. Any more questions? Okay, while waiting for the other question, I have a question for Professor Chin. It's a more broader question beyond what you presented today. You have helped reshape how we view death through the Silent Mentor program. So in your experience, how this has transformed the way medical students, undergraduates, postgraduates view, like end-of-life care and patient empathy and so on, the humanistic aspects?
That's a very good question. So we publish data at the medical educations that shows that our Silent Mentor program indeed can increase the empathy level of our medical students who join the program throughout the workshops. And this has been proven on statistical analysis. And not only that, we also noted that our donors actually appear to have a better quality of life towards the end of their journey before they pass on, because I think maybe psychologically or spiritually they feel that they have contributed something to the society. At least there is some worth and a kind of meaning towards the end of the life. So at the moment, we are actually setting up a research studies to look at this, how altruism alter the concept of death for our terminally ill patients. So I think this will let us understand more about why the program itself works for cancer patients as well. Why more than 50% of pledges actually cancer patients. And I think clearly we look forward to see how the rapid tissue donation program will work in Malaysia, and how many of them will actually come forward and donate their tissues. Because there is a lot of taboo, because you have to reach us, you have to harvest the tissue within 4 hours. So a lot of the Chinese will think that I can still feel the pain after I died. And so there may be a lot of hesitance. And also the family members as well. We have to get the support of family members. Because our bottom line is that if any of the family member object, then we will not pursue the donation. Yes.
Very nice, Prof. Chin. Any more questions? I have a question for Dr. Akhmal. Oh, I see a question. Okay, please.
Hello, good morning. I have a question to all three of you. So just now from Dr. Akhmal's presentation, you mentioned that the increase of discovery of mutation in cancer diseases, and also so in response to that, so there's an increase in ADC and also the radioligand. I wonder what's your opinion on gene therapy, because in my experience, I'm from FRA, I'm from biologic section. So in my experience, we don't really see the increase in gene therapy, for diseases, although there is an increase in the discovery of mutations in so many diseases. So.
I think to answer your questions on that basis, I'll start with congratulations for MPRE for bringing out the first in human to cell gene therapy. That's a great yardstick in terms of scientists that is managing these. Specifically, I can't answer with regards to the how it works in the cancer cells. But having said that, that is another opportunity to understand cancer cells itself. in terms of how we can delete that that part of the gene, what do you call it, coding, to replace a new cell and gene for that matter. So that's probably the idea of the insertion of new cell replacing all these cells that is not available, that is not causing trouble. So I think that is the fundamental of it. But what we can see here, 32%, 1/3 of the new development or new clinical trials are on that specifically last year alone. And this was reported by IQVIA. So that means this would mean there will be more studies will be going towards that area, and hopefully there will be a scientific explanation that can satisfy why that cancer cells react to the cell gene. Do you have anything?
I think at least from a scientist perspective, right, I mean, we always ask the same question. We have CRISPRs, we have so many different editing methods that can help supposedly improve the healthcare, but why is it not being taken out? So I think it is easy to prove things that are there. So we know that this gene therapy, the knockout, these particular genes will cause response to the tumor cells. It's easy to prove that. It's easy to convince people that it's work. But you can't convince people what is not there. That means what is the effects on the normal cells, what is the effect on the immune cells, what is the effect on the entire ecosystem. There isn't enough evidence or proof to actually showcase that it's only working on the tumor cells, the tumor will be fine. Right? It's always about convincing the patients which drug to take. I mean, there are so many drugs out there. Right? It's really about getting the best drug, the best dose to the patients. And this is why I think gene therapy has a lot of uncertainties that has to be ironed out before it can be eventually launched to the market.
I have to stop the session, unfortunately, because we are reaching closing. So any questions, please carry forward. We have a coffee break at 10:30 onwards, so you all can continue asking them if they don't mind. Okay, and we would like to reach the end of the session, so we are presenting tokens for our esteemed panel.